Our next speaker is Professor Ron Fan. He's the Harold Hodgkinson Professor of Biomedical Engineering at Yale School of Medicine. And he will talk today about the special multi-omics sequencing via deterministic barcoding in tissue. So thank you very much.
Yeah, thank you. Thank you for having me. I'm gonna go ahead and share my screen first. All right, can you see my slides?
Yes, we can.
Okay. Okay, I'm gonna minimize my own camera. All right, yeah, thank you so much for having me. This is a great opportunity for me. I'm really honored and also very humble to have this opportunity to share my work. I'm not a kind of epigenetics researcher, and this is a great opportunity for me to learn a lot of exciting work in this space. And also, I'm new to aging and senescence research, but I decided to change my title of my talk a little bit to say some preliminary work we have been working on towards the study of aging and cellular senescence using spatial multi-omics, in particular spatial epigenomics technologies. So it's going to be technology focused. But I hope I do share with you a little bit new insights, potential and the new technologies can reveal. All right, it's my disclaimer. And so my kind of research program in this space is largely driven by kind of grant we received about a year ago from the Senate Consortium to mapped out the human lymphoid tissue lymphoid organs and where the senescent cells are and how the senescent cells might impact the tissue local microenvironment through the SASP factors and the cell-cell interaction, and how does that eventually change the physiology or pathophysiology. And so the four different organs we're interested in, I think, bone marrow, thymus, and tonsil, and the lymph nodes, and in particular lymph nodes. And as I depicted here, the lymph nodes are actually quite interesting, the secondary lymph organ that filters on foreign particles. But unfortunately, I think the organ does not have the detox function. So over the time of the entire lifespan, basically you'll see accumulation of the stress signal and the toxic stress in lymph nodes, and you see the activation of those fibroblastic retinal cells, and some of them might become senescent, might produce the collagen extracellular matrix, and eventually alter the normal physiology of T and B cells, sort of the organ responsible for example, if you get a Moderna vaccine, so the B cell training, B cell maturation, selection, and eventually the development of the antigen-specific B cells that occur in the lymph nodes. And if you are kind of older versus younger, and so definitely this gonna accomplish change the development of adaptive systemic immunity. So this is a kind of very important organ, I think, all the lymphoid organs, and that have a systemic impact on human physiology across almost all other organs in our body. All right, so the technologies we have been kind of using over the past couple of years to look at the single cells really revolutionize how we study biomedical and biological questions. But we do realize, I think the field realized, okay, that's not enough. Oftentimes you don't have the spatial information over there. So I think the technologies that emerge over the past 7 to 10 years to address the challenge, sort of the spatial resolved single cell omics analysis. There are two major categories. One is based on imaging, such as MERFISH and SIGFISH. So you can image the gene, the transcripts, and over multiple rounds, and through the combinatorial barcoding approach to reach almost entire transcriptome. you can also do repeated cyclic imaging of proteins and other things. But what we are interested in is, can we use the high throughput NGS sequencing to computationally reconstruct the spatial gene expression or potential other omics as well? And so this field started about six years ago, when this landmark paper was published by Professor Joachim Lamberg in Science, talking about how you can use DNA microarray to capture messenger RNA released from the tissue section. You place on top of the glass slide. You place on top of the DNA microarray, but each DNA spot gives you the spatial zip code. You can incorporate into the cDNA if you kind of synthesize the cDNA on a glass slide in situ in the same sort of the location. But that spot size was pretty big. I think the excitement wasn't that high when my colleagues read this paper, but that's just the beginning, very, very beginning of the whole story. And the field evolved so much over the past couple of years. I think, for example, the 10x Visium and also the SLICE HDST 2 to 3 years ago already demonstrating the spot size can be much smaller, close to single cell level, which is about 10 micron, 5 to 10 micron. And last year, 3 technologies further demonstrate you can get the spot size down to 1 micron or even smaller. Really, it's kind of approaching the single cell or even subcellular spatial transcriptome sequencing. So they all follow actually exactly the same fundamental principle developed by Professor Joachim Lamberg, which is you capture messenger RNA on a barcode solid surface and do whole transcriptome sequencing. So what we came up with is kind of total opposite. Instead of capturing messenger RNA, we send the DNA barcodes in to tag the transcriptome, but you can also tag many other things. In particular, what I'm going to talk about today is you can tag those epigenetic features, either chromatin accessibility or chromatin modification. So how the technology works, I think that has been published a number of years ago. We use this very simple microfluidic device to deliver DNA barcodes over the surface of the tissue. The first set of DNA barcodes give you sort of the row barcodes, and each of those has the oligo-DT that binds to the poly-A tail of the messenger RNA or the poly-A tail of the DNA-derived antibody tags, such that you can simultaneously measure proteins and transcripts. But that gives you only one direction, right? So if you want a 2D spatial mapping, you need another direction to introduce a second set of DNA barcodes, in this case B1 to B50. And then you ligate them together, and the intersection, each of the kind of intersections has a unique unique combination of the barcodes and the B. So together you are able to kind of map down the biomolecular information, either transcriptome or protein, pixel by pixel across the entire tissue, as demonstrated here. So when we published that paper, I think 2 years ago, the spatial transcriptomics was highlighted as the method of year by Nature Methods. And in the same issue, they also highlighted our work saying multi-omics sequencing goes spatial. And so just maybe 2 or 3 slides showing when we talk about multi-omics that time, that was kind of panel proteins and a panel of like 22 proteins and a whole transcriptome. You may ask, okay, how many proteins you can look at to get a truer kind of proteome scale versus plus transcriptome scale co-profiling. So recently we have a paper just accepted in Nature Biotech that demonstrated the hundreds of proteins simultaneously mapped out with the whole transcriptome, and also demonstrate across a variety of different tissue types, spleen, colon, intestine, kidney, from mouse, but also from human samples. For example, here there's a tonsil. We use there's 300 proteins plus the whole transcriptome to map down this particular region right here, you can see probably there's a B cell follicle, there's another B cell follicle from all 300 proteins. When you perform unsupervised clustering, you can actually see the all the major clusters in the UMAP, but this is spatial, so you can go back to see where those clusters are located. And so here, you can see there's a B cell follicle, another follicle, and there's a T cell zone. Those are the T cells. And in the follicles, you can see light and dark zones. There's an epithelial crypt and a lot of bluish extracellular, extrafollicular zones. So the transcripts give you very similar spatial pattern, but somehow the protein gives you much more kind of distinct spatial features. People often ask me, I think, a protein versus transcripts, which one gonna do better job to identify spatial features? My answer is I don't know. Just want to let you know, even though we sequence, we profile 300 proteins using 10% of the sequencing length, but actually the sequencing depth per protein is 10 times higher than the sequencing depth per gene in the spatial transcriptome data. So you can look at individual proteins, for example, in the B cell gene and this follicular dendritic cells. You can also look at a functional proteins such as IgM, IgG, IgD, associated with the kind of germinal center B cells and more mature IgG producing B cell already migrate out of the GC, and already migrate out of follicle, and the naive B cells just act in the blood vessels. They're right here. You can also look at the CD4, CD3, those T cell markers, and also some kind of lymphocytes that are kind of outside the follicles. So we did a benchmarking kind of against the immunofluorescence imaging. For example, the serial section, the adjacent tissue section, you can do multiplex immunofluorescence and compare. You see very, very similar spatial distribution of the same protein. You can also compare or benchmark against the single cell CITE-seq when we have the two data sets combined, our spatial CITE-seq pixel by pixel on a single cell CITE-seq data points. When you combine them to perform unsupervised clustering and present the UMAP, you can see for every single protein where they are, okay, in a UMAP space, you see very strong concordance. For example, CD3 right here, and the CD20 is very small corner right here from both the single cell and the spatial CITE-seq data. So now we are trying to apply the spatial CITE-seq to the cellular senescence research. There's some preliminary data we got from human liver. We look at one of the classical senescence markers, P21. We also look at three others. So using the immunostaining, you can see where the P21 is relatively enriched in this liver specimen. From the same sample, we did a spatial CITE-seq. We can identify basically all the proteins, differentially expressed the proteins, but also the entire transcript. Now, what I'm showing you here is the 4 senescence markers. They show pretty kind of consistent pattern where they are located. They are enriched in the tissue. They're all right here. For HMGB1, I think it's the gamma H2A, and a P16 and a P21. I think a classical senescence markers, but ideally you can look at a whole entire panel on the signature of the cellular senescence rather than just 4 markers, which we're able to do, and there's still ongoing work to further dissect the heterogeneity of those senescent cells rather than just, okay, there are senescent cells. Okay, we want to understand any difference among the senescent cells and how they interact with the local tissue environment. Okay, finally, next half of my talk about the epigenetics, how to kind of spatially map down epigenetic kind of chromatin accessibility or epigenetic histone modifications. The first is global analysis of the chromatin accessibility using ATAC-seq chemistry. But interestingly here, we can, after we direct process the tissue using the TM5 transposes, and insert the DNA oligos to the open chromatin region. If you do bulk ATAC-seq, actually, you can go ahead and digest the tissue and run sequencing. But in our case, we did 2 more steps to add the barcodes A and the B, the horizontal and the vertical, and then you are able to map down sort of the chromatin accessibility pixel by pixel. I demonstrate here, this is an embryonic mouse liver, and you can see all the pixels in the embryonic mouse liver. Every single pixel, we have the genome-wide chromatin accessibility sequencing data. And if you aggregate them to produce the pseudo bulk data, and that turns out to be in very strong concordance with the ENCODE reference data. I believe this is from Professor Binzen's ENCODE reference data in the mouse embryo. And so now we look at the human tonsil or lymph nodes again to see how in the lymphoid tissue, sort of the different chromatin accessibility, the different sort of the chromatin accessibility can be mapped out pixel by pixel in the particular region of this tonsil tissue. So we use relatively low resolution clustering. Low resolution here means you don't try to get too many clusters, just try to identify the major regions or major clusters. So now we see 3 major clusters. So one corresponds to the follicle, the other extrafollicular region. The third one is sort of the epithelial crypt. So now in this case, you are no longer limited to the 300 proteins you are able to put together. You can look at whatever markers you are interested in, or whatever genes or the sort of the accessibility of the genes, right? So we look at the B cell gene, for example, CD10 enriched, supposed to be the marker for germinal center B cells. You see the accessibility is exclusively enriched in a follicle. But you can also now look at those master transcription factors, for example, PAX5, known to be master transcription factor for germinal center B cells. You see that's strongly enriched in GC. For the T cells, you can look at those kind of T cell markers, but also the master transcription factors, for example, BCL6, which is a transcription factor for germinal center B and also follicular helper T cells. You see that's exclusive expressed or accessible inside the follicles. What is interesting here is the FOXP3. If you study T cell biology, you know this regulatory T cell master transcription factor. We see extensive FOXP3 chromatin accessibility in the extrafollicular zone, but you don't really see a lot of FOXP3 T cells, regulatory T cells. So our understanding is, so basically the nature actually very smart, our body is very smart. In the tonsil, we always have those FOXP3 epigenetic reader cells or epigenetic primer cells, but they don't have to be Treg per se, okay, but they have the potential to quickly turn themselves into Treg if they see the local, for example, hyperinflammatory response. So they are always ready to rescue if they need to do that. You can also look at the pseudo-time cell differentiation all the way from, for example, naive B cells to germinal center B cells. You can look at the differentiation trajectory, very much like how you do a single cell differentiation trajectory analysis. But the difference here is you can look at where they are spatial in this tissue. So, okay, now what we try to ask in this tissue sample is, can we look at where those kind of senescence markers are in terms of their epigenetic state? In immunosenescence, I think in the P16, P21, again, should be kind of elevated in the senescent cells. And many other things you can look at as well. I just show you there's 2 P16 and a P21. So now we do see a lot of those P16, P21 epigenetically positive or epigenetic accessible cells in the extrafollicular region, and also relatively consistent between these two across the entire tissue. So where you see P16 positive, you see a relatively high level of P21. So I think following a fantastic talk from Professor Binzen, I think we all appreciate the aging is strongly associated with the epigenetic alteration, and the epigenetic clock of aging has been widely kind of appreciated as very important aspect of the aging mechanism. But I think we also believe the devil is really in the details and the single cell level in the tissue context. And so in addition to global alteration of the epigenetic sort of epigenetic state, and can we really kind of map out the alterations pixel by pixel in the tissue and the single cell level to really understand the mechanisms. So we were also able to look at a specific histone modification rather than just a global accessibility of the chromatin. And so, as demonstrated here, we look at 3 different histone marks in the mouse embryo, and even without doing multiplex cut and tag, you are able to kind of compare the spatial distribution of the different clusters across 3 different histone marks to kind of come up with how the different histone marks work together, either synergistically or in some kind of mechanism to collectively control tissue and the cell type development. So here I just show you one, again, show the time from radioglia to accessory neurons, and in between we have the post-mitotic premature neurons. So in this case, you see on the sort of the genes controlled by this particular histone mark. Okay, this is not just the cell type differentiation trajectory, but this really tells you how this histone mark controls this differentiation trajectory, or somehow involved in this differentiation trajectory, and you can directly visualize that in the tissue. So we focus on this particular region, but actually see many other activities in other regions of the mouse embryo as well. Not a surprise, you see radioglia largely in the ventricle. Once they are more post-mitotic premature neuron towards the accessory neurons, they migrate into the cortical layers. And now you see, actually this process is kind of controlled or maintained by this histone modification K4 trimethyl. So since we are able to do whole transcriptome and the epigenome, the question is, can we do both? And it turns out so many people are excited about this capability to co-profile epigenome and transcriptome. So in our approach, it's not very difficult to combine spatial ATAC and a spatial transcriptome, or spatial cut and tag and a spatial transcriptome. Just give you one example here to describe the workflow. And so once you fix in the tissue, and you'll use the TN5 transposase to process the tissue first, that's invoked. You don't have to use the microfluidics at this step. And afterwards, you introduce the primer to kind of oligo DT primer to probe on the transcripts and perform RT on the tissue. After these 2 steps, you add the barcodes A and B, but simultaneously co-index the transcriptome and the epigenome. So finally, you'll collect the cDNA and collect the genomic DNA. You'll prepare 2 sequencing libraries, and so now when you sequence both, you are able to link them together in the same tissue section and pixel by pixel across the entire tissue slide. So this is the data from mouse embryo, and so you can look at this kind of epigenome part of the data, and also the RNA part of the data. From each of those, you are able to perform your independent unsupervised clustering. So you do see pretty strong concordance. I think either modality allows you to identify major tissue types or major spatial features. The transcriptome gives you a little bit more, and that's not a big surprise because I think the epigenetics, they can further diverge into different transcriptional states because there are many other regulatory layers kind of above the chromatin accessibility. But you can also combine them to do kind of more precise, more reliable tissue or cell type identification. So this kind of joint analysis of the spatial ATAC and RNA. So in this case, you are able to look at the shorter time. You can look at individual genes. I just give you one example. That's the radioglia differentiation towards the most mitotic premature neurons. And so you can look at the sort of the differential expressed genes in this case in relation to the shorter time differentiation process. And so not a big surprise in many genes. Actually, what I'm showing you here is not just all of them. Actually, if you look at all of them, most of them, I think, do follow sort of the chromatin open first, and then later you see transcriptome transcriptional activity showed up. Okay, that's kind of what you anticipated. For example, here for those genes that they are associated with kind of neuronal developmental process. But interestingly, we see a group of those genes kind of transcriptional expression goes down, but the epigenetic accessibility remains high. And my collaborator Gonzalo Castello at the Karolinska Institute, when he looked at it as well, he said, those are my favorite genes associated with oligodendrocyte or oligo kind of OPC, oligodendrocyte progenitor cells. So the implication here is you actually have a lot of cells that already kind of differentiate into neurons. They are post-mitotic premature neurons, but they maintain their kind of chromatin open for oligodendrocyte lineages. So they have the potential to trans-differentiate them into oligodendrocyte if necessary. So that's kind of interesting finding from this data. So we did a spatial chromatin accessibility, chromatin modification, histone modification, whole transcriptome co-profiling across 3 different mouse tissue sections. For example, here, I think you see the from this K27 trimethyl, you get a transcriptome. From 2 other histone modification, you get a whole transcriptome. So it turns out this kind of like the technical replicates. And from this data, you do appreciate how strong concordance between the technical replicates in terms of the transcriptome. But from the epigenome, you see some differences, although you do identify the major tissue types, tissue regions in the brain, in this case, almost entire kind of hemisphere of the mouse brain. This is P22 mouse brain. So you can also integrate the single cell sequencing data, single cell cut and tag or single cell transcriptome to identify all major cell types well annotated. And now you use the label transfer to find out where the different cells are in every single pixel, and then you can go back to visualize the cells directly in the tissue. Since you have the epigenome and transcriptome, I think an interesting analysis you can do is no correlation between them, right? So I think this is suppressor mark. You can look at sort of the gene histone activity versus the expression of the genes, and not a surprise, most of them are kind of anti-correlated because this is a suppressor mark, but you do see some outliers, and outliers are not a big problem, but when we publish the technology paper, sometimes that's a little bit challenging, but I really believe that's where the interesting biology going to emerge. When you look at this 2 other histone marks, they are either a kind of promoter or promoter enhancer gene activity marks. You do see kind of the genes are in either first or third quadrant and very few outliers. So now we try to look at in this specific histone mark and across all different regions. Since this is spatial data, you can look at a pixel by pixel. Also, you can aggregate the pixel in a particular region, for example, all different regions here to perform this correlation analysis in a region-specific or cell tissue type specific manner. So what you see here is that in a corpus callosum, the histone mark, but our interest is really right here in the corpus callosum. You see this particular gene associated with neuronal and synapse development. I think strongly in high in this histone mark activity, but this is suppressor mark. You see the gene expression is low. I think conversely, you also see the activity for two other histone marks and also ataxic chromatin accessibility is extremely low here in the corpus callosum. And a MAG2, MAG is a gene for oligodendrocyte and oligo-OPC lineage, and you see the suppressor mark activity is low right here in a corpus callosum, and as a result, you see high gene activity for two other marks and also for ATAC and also across all different samples, the high expression and a transcriptome level. And so interesting, these two are the outliers. I just show you one of them, PLP1. You do see kind of high activity of this K27 trimethyl, but interestingly, the gene expression is also high, although this is a suppressor mark. So I think in this case, that means this expression of this gene is controlled by many other or maybe collected by different histone modifications or other epigenetic, and this is so where the more interesting biology you can figure out. Okay, just to try to wrap up by saying, although we are very, very proud of those spatial epigenome sequencing technologies, but the real power might be the data you can produce across different modalities and fully integrate them together, in particular the imaging plus the sequencing-based spatial omics. So here, what we try to do is to, for example, in the lymph nodes, to map down 45 different protein markers to identify all major cell types. This is data from the Akoya Codex multiplex immunofluorescence imaging across 45 different markers. And you can perform the unsupervised clustering of all the 45 markers to identify major cell types. This is all different clusters. You can do your own annotation to find out which cell is which in every single pixel. But now what we are trying to do is from exactly the same tissue, we can put our device to map down entire transcriptome or entire epigenome. But in this case, you know exactly what you have in every single pixel. This is the grid, is 100 by 100 or 50 by 50, but every single pixel here you can see exactly what cell you have. I think at this resolution, we have only like a 1, 2, 3 cells per pixel. It's very easy to really kind of deconvolve or decompose in this case, not a blindly, but actually you know exactly what cell you have. You can do your deconvolution in a much more precise, much more accurate way. So another layer, I think one of the questions to Professor Binzen's talk is metabolic state. And we're very excited about this aspect as well. And turns out some technologies are relatively non-destructive. For example, in collaboration with Professor Lin Yanxi at the UCSD, we try to combine the stimulated Raman spectroscopy to image metabolic activity. For example, lipid unsaturation and an oxidation, oxidation activity by looking at these two metabolites, ADH and flavin, and many other things. But interesting, afterwards, the tissue is still intact, and we try to do spatial omics sequencing, either epigenome and transcriptome. But afterwards, you can really combine, integrate data altogether exactly on the same tissue to really link the epigenetic states, in particular in the senescent cells, where they are, what they do in terms of metabolic, in terms of transcription, in terms of epigenetic, altogether on exactly the same cell. That's our vision. All right, so briefly summarize, we develop a suite of spatial sequencing technologies based on this deterministic barcoding in tissue to look at transcriptome proteins and the epigenome, potentially other things. And the real power is really to integrate them seamlessly with the imaging technologies. And what we do in the Senate consortium is to generate such data across different human lymphoid tissues and try to understand the impact of those senescent cells on normal human physiology, aging, and maybe other diseases as well. All right, I would like to thank people from my group and amazing collaborators from the brain research. Actually, Gonzalo Castillo Branco from Karolinska Institute has been a terrific collaborator for years, and now within the Senate, I look forward to the collaboration with many colleagues within the Senescence Consortium, including Peter Bing and many others. Thank you very much for your attention. If time permits, I'm happy to address a few questions.
Thank you very much. It has been just a fascinating talk. And maybe while we wait for a couple of questions, I'm going to start first because I have a curiosity. I think I was just wondering in your experience, which kind of modality is more stable for FFPE samples? Because with RNA, we know that the quality of the samples, if they are very old, it will not get very good quality, but maybe there is another modality we can rescue those archived samples?
Yeah, this is really kind of on top of my list this year to get it worked out. Yeah, I would say that how robust it is, it depends on the sample first. Okay, if you have no RNA left in the FFPE tissue specimens, and no way you can get any transcriptome, but maybe you can get an epigenome. That's what we're trying to figure out, whether now the genomic DNA might be even more stable than RNA, and the intuition is yes, but we need to kind of produce data to demonstrate. But if I had a good sample to start with, which platform might be relatively robust, I would say maybe probe-based are going to be relatively more robust. I think the 10x Genomics has the FFP Visium solution, but that's basically a fish. That's kind of NGS radar for fish. That's completely probe-based. If you are interested in the biology of RNA rather than just know an expression of those genes, you still need to do base-by-base sequencing. So now, so I think our technology works, but I'm still trying to figure out why sometimes that works, why not, and how to kind of define the tissue quality matrix to get a work. And there are several tricks. I believe you're going to see amazing breakthrough in this, in the FFPE spatial omics, including epigenome transcriptome.
Perfect. Thank you. Gary, I think I see your hand.
You do, Celia. First of all, Rong, a fantastic talk, a ton of really good data. I have a question here from one of my colleagues, Michael Garbati. Mike, if you would like to ask a question.
I'm Mike Garbati. I'm a product manager for our services. And my question is, I think you answered it on some of your slides. How many cells are in a 50 micron pixel? And is that cellular density, I think, will vary across different tissue types. So how do you deal with that?
Yeah, absolutely. So I think if you are looking at human brain in a kind of neuron-rich regime, so not a microglia, even 50 micron, you might get a lot of single cell data points from 50 micron. So we don't really do 50 micron a lot. We do about like a 20 micron, 10 or 20. Even for lymphoid tissue, 20 micron, you do see a lot of single cell data points, not all of them. So on average, for 20 micron, probably one to 3 in those tissue types. But I think in the embryo, definitely you have more cells. For 20 micron, it's probably 4 to 5 on average, but it depends on where you're looking at, what tissue organ. Like a cardiomyocytes, even probably don't, even 20 micron, you'll probably get a half a cell because those are huge. Yeah.
Thank you. I see another question from Bing.
Hi, Rong. That was fantastic talk. Very impressive, truly outstanding. I just have one quick question about your slide showing elevated expression of the P21 and P16 in, I think, one of the tissue is at liver. I wonder whether you were able to demonstrate that this elevation is spatial specific for the same cell types. In other words, it's not just for cell type that show this elevated senescence mark, but also it's highly non-random in space.
Yeah, this is a great question. So even the same cell type, right? So the senescent cells are somehow clustered in a particular region due to, for example, maybe local stress. That might be in a case in like a lymph node, because that's where the foreign particles kind of localized in a lymph node might trigger the toxic stress and then serious senescence. Yeah, for the liver sample, that's very, very preliminary. We will follow your suggestion in our future data analysis. So that sample is not a normal human donor liver. That's a NASH. So that's probably why you see a lot of those P16, P19 positive cells, both transcriptional and that epigenetic chromatin accessibility. Yeah.
Great, thanks.
Yeah. Also back to the previous question, I think the how many cells and how to deal with that. I think I answered your question, how many cells, but how to deal with that. I probably just want to kind of share what my computational biology friends told me. I thought that's a problem, that's a challenge I cannot handle, I cannot deal with. But my computational friends said, no, don't worry. So go ahead and produce your data, we will take care. And so I think many computational pipelines already developed to do cell type deconvolution and decomposition, and that worked out quite well. And in our case, we do very simple, just combine the spatial data pixel by pixel and the single cell sequencing data. We put them together as one data set to perform on a clustering analysis and then label transfer. So in that case, we can identify major cell types, not all different cell types in each pixel. Yeah, but so this is sort of kind of blind deconvolution. But as I alluded in the end of my talk, I really believe in the real power is if you can image exactly the same tissue, you know what cells you have in every single pixel, and that's gonna help you a lot to kind of disentangle the different different cells and to produce the truly single cell spatial omics from this pixel by pixel sequencing. We may not be able to get it to subcellular, but it's totally possible. And we are almost there to generate sort of the truly single cell spatial omic genome-wide spatial data.
Thank you. I have another question from the Q&A, more also technical. It's from Amber Alley. She's asking, how is the segmentation of the cells performed? Which algorithm or software prior the spatial analysis?
Sorry, can you say that again? How was the tissue prepared?
How is the segmentation of the cells performed before the spatial analysis?
Okay, segmentation. That's something we're not very good at. Okay, the imaging, sort of the imaging-based spatial omics, where we're burning cache on Illumina sequencer. But I do want to kind of bring like a Codex and a Murphy's exactly on the same tissue, but afterwards definitely gotta do on a cell segmentation. We haven't done any Murphy's, but that's kind of on my radar. And for the Codex multiplex immunofluorescence, I think we're using a built-in software from Akoya to do cell segmentation. I would say that works fairly well in the tissue types we're interested in, those kind of lymphoid tissue, lymph nodes, tonsil. But maybe for different tissue types that they do need to kind of further optimize a little bit. I think Akoya has extensive data from tumor microenvironment, tumor immune microenvironment, that kind of spleen, tonsil. So that's the type of tissue that the segmentation algorithm works quite well in Akoya Codex. Yeah.
I have another question. So it's regarding the cost and the high throughput, right, of spatial transcriptomics, whether it is for large cohort of samples or biobanks. So the DBIT-seq is cheaper compared with other platforms, but still is very laborious, needs a lot of time to perform the experiments. Do you anticipate that in the future will be any kind of automation to do the spatial transcriptomics in a more high throughput?
I don't think the throughput. So I think the beauty of the NGS-based spatial transcriptome or spatial omics, broadly speaking, is potentially it can be extremely high throughput because you already decouple your sample prep and your radar, right? So you just put the library in the Illumina sequencer. So you're totally right, but the question is now I don't see a lot of research laboratories want to do that extremely high throughput, but now the consortium like a BICAM Brain Initiative and the center, so we are being tasked to develop higher throughput, and it's totally durable. But yeah, think about afterwards how much cash you need to spend on sequencing. So nowadays, okay, for 10x, I know the kind of 4 samples, 4 tissue section per round per slide. If you look at the tissue, the glass slide, I would say they can easily put on 8, right? Because they put 4 on just one side. And also for our device, so we haven't done that much kind of multiple tissue section per slide. But turns out a group in Germinal, is Immunofluorescence, they adopted our technology. They developed something called a DBIT-seq, so that they are able to have like a 9 different tissue sections per glass slide, and simultaneously run all 9 tissue sections. Yeah, that's without robotics. If you use the robotics to load all the reagents, I would say it's now difficult to achieve the throughput, the sample processing throughput, and the scale of like 50 tissue sections per day. Okay. That's totally achievable. Yeah.
Okay. Thank you.
Celia, do you want to ask one more question for Rong and then move on to the roundtable, or?
Okay, let me double check the Q&As.
Was there one left? I think there's one from Sarah Fries. Yes,
that one. Maybe I can just read it from Sarah Frias. Said, okay, amazing talk. I'm interested in senescence in patients with bone marrow failure. Is it possible to study aging with some of these methodologies in blood and bone marrow smears?
Yeah, this is great. This is exactly what we're interested in. So I think a bone marrow is really the tissue I'm most interested in, but most difficult as well. I think any spatial study of bone marrow, if that's no bone marrow biopsy, you got to do decalcification. You'll damage a lot of RNA. We still don't know whether or not the genomic DNA is still okay, and we're working on this. We have some preliminary data from spatial multi-omic sequencing of bone marrow. It is working, although the transcripts capture, the number of transcripts capture a little bit lower. Yeah, stay tuned. That's what I can say. And yeah.
Perfect. Thank you.
Yeah, I see one question in the chat box. Sort of, can you also look at non-coding RNA? Actually, yes, and we have beautiful data from spatial small RNA and a kind of 1,000 to 2,000 microns mapped out pix by pix. So that's totally durable.